Hello everyone, and welcome to our online lecture series for plant biology. Today, we tackle probably one of the most fundamental and most important concepts that we'll cover in this class. After all, this is a class about plants and what plant function is more important than photosynthesis. Now, I will warn you, one of the reasons why we do this two-lecture series, it's a two-parter, on photosynthesis early in the class is because it also covers probably the most complex molecular aspects that we will discuss in this class. I like to include it up front because that way you can get a feel of the highest bar that you'll have to jump over. You feel the confidence that you need in order to tackle this complex but very cool concept. And then it gives you an appreciation for how much more complexity there can be to some of the topics that we go over later in the class. So yes, we will go into such minute detail on photosynthesis that we will be discussing individual protein function in the different compartments of the cells of the plants. But before we get there, I'd like to discuss some of the basic background on how we even figured out that photosynthesis is a thing in the first place. It really puts into perspective how scientists work in order to slowly but surely uncover the puzzle pieces to put together the entire picture. So, dates here are not important, but I will kind of introduce the scientific experiments that got us to our photosynthesis equation. The first set of experiments were done by an individual named Van Helmont. He was a priest slash or a religious individual, and also a scholar, as many folks were in the late Middle Ages, early Renaissance. And Van Helmont had this hypothesis that plants, like humans, had to eat something. But he liked to observe plants and noticed that they did not eat any animals that came by, but that their roots did grow in the dirt. So Van Helmont had this idea that plants eat dirt. This was his hypothesis. And he designed a simple experiment to test this hypothesis, an experiment that you could do in your own home. He had a willow tree that he weighed and got a number of pounds of it at the beginning. He uprooted it from the ground and measured its entire weight. He also had a pot of soil that he measured how much it weighed. Now, he started this experiment in the late springtime before growing season, or at just the beginning of growing season, and he had an original weight of things. And he logicked that whatever weight that would disappear from the soil would have to reappear in the plant, that the plant would gain as much weight as the soil lost. Seems sensical, right? Fairly logical. Now, he knew that plants also needed water, so during this whole experiment, he was watering the plant. Y'all will be familiar, hopefully, with the fact that the chemical equation for water is H2O. Granted, Van Helmont did not know this at the time, but he was watering the plant regularly. And after the course of 2 months, what Van Helmont did was he uprooted that entire willow and got a second measure of how much it weighed. And the weight afterwards had increased. After all, the plant had grown for 2 months. And then he took all of the dirt in that pot and weighed it out and noticed that there was no change. So Van Helmont's hypothesis that plants eat dirt was proved to not be the case. He realized that none of the dirt disappeared, or at least very little of it, to the point where he couldn't really measure it, and therefore the plant could not be eating the dirt. Instead, Van Helmont logic that the one thing that he was adding through this entire process was water. And therefore he concluded that one of the important foodstuffs for plants was water, and that plants were able to grow and produce energy through the digestion of water, or H2O. Now, he kind of missed the mark, unfortunately, right? We know that water doesn't provide you calories. Today, you can see that by looking at the nutrition label of water bottles. No calories in there, right? Water is an essential component of many different chemical reactions, but it is not a source of calories for us. In order to really understand how plants were able to get calories from things or generate energy, we need the experiments of several more individuals. And while the scientific record of how many people contributed to photosynthesis is kind of long, I'll only introduce 2 individuals in that process. An individual by the name of Priestley and an individual by the name of Ingenhousz. Now, these 2 did not work together, but they built on each other's experiments. Priestley conducted experiments where he showed that air was essential to supply, or that the energy in air could be depleted, or the resources in air could be depleted. He did so by having a glass jar and a candle. This candle would be lit. Now, in this glass jar, he noticed that eventually the candle flame would go out. He figured, maybe something weird is going on there. Well, he also started to see if something was different about the air in that glass jar when the candle flame was going out by putting a little mouse in there. That's as good of a drawing of a mouse as you're going to get from me. Maybe some toes, right? Now, what he noticed is that when the candle flame went out, so did the life of the mouse. He was figuring out that there is an essential component to the air in this jar that we would later call oxygen, and that the burning of the candle would make it so that the oxygen in the jar would become depleted, and the mouse would therefore die, and the candle would no longer burn. Ingenhousz took this experiment and added another variable to it. He noticed that if you put a bit of plant life in here, that the candle would burn longer, and that the mouse would live longer. His conclusion was that the plant was somehow making oxygen. Pretty clever, right? Now, of course, the plant would not be able to keep up with the amount of combustion occurring, and the mouse would eventually die, so rest in peace, many mice who've died for plant experiments, not even mentioning how many mice have passed for countless numbers of animal experiments. But through these experiments, we see all the different components of the equation for photosynthesis. We already know, thanks to Van Helmont, that water is an essential input. We also know that oxygen is one of the outputs of photosynthesis. It is a byproduct of whatever the plant is doing. And then we required a couple more chemists alongside Ingenhouse and Priestley to put together that carbon dioxide, or CO2, was going into the plant, and that sugar in the form of glucose was the chemical energy that was being produced via photosynthesis. Now, we all know today, if you knew one thing before this class, was that an essential energy source for photosynthesis was sunlight. You cannot produce sugar without sunlight. Plants cannot live without sunlight, and plants are able to convert sunlight into chemical energy. And that's the essential crux of what photosynthesis is doing. It is converting light energy into chemical energy. Chemical energy in the form of sugar, a carbohydrate. And you'll know from reading the back of your cereal box that carbohydrates have the calories needed in order to keep us alive. Always start my day with a bowl of Raisin Bran Crunch, full of plenty awesome carbohydrates. Definitely my favorite cereal out there. That give me the energy that I need in order to function and record these lectures. So this is the experimental background that we need to understand in order to build on our fundamental understanding of photosynthesis. We have the inputs of sunlight coming into a plant, carbon dioxide being breathed in through the leaves of a plant, water being absorbed through the roots of a plant, and with those three inputs, we have the outputs of sugars being made and oxygen being breathed out. It is pretty cool that plants are breathing in carbon dioxide and breathing out oxygen, because you will know that you as a human, through the process of cellular respiration, will breathe in oxygen and breathe out CO2, therefore creating a loop. It is no coincidence that photosynthesis and cellular respiration create this loop of renewable energies. Of course, cellular respiration is happening in the mitochondria, the powerhouse of the cell. Now, we need to take a deeper dive into what exactly is happening in the organelle where photosynthesis is taking place. That organelle being the chloroplast. But we need to talk about where these chloroplasts are found, because not everywhere in a plant produces sugar via photosynthesis. Just like in us, we have specialized organs or tissues for movement and filtration and thinking, like your brain. Hopefully you are indeed using it right now. Plants have specialized organs and tissues for different functions. The organ specialized for photosynthesis is, of course, the leaf. The thin green material that performs so much sugar production. Now, under a microscope, as we will see many, many, many times in this class, a leaf has different layers. Of course, there is an upper epidermis and a lower epidermis, the skin layers. But you also have these two layers that are photosynthetic, highly photosynthetic. Collectively, they are called the mesophyll, or the middle leaf in Greek. I think that kind of makes sense, right? Look at where these two layers are. They're between the upper and the lower epidermis. Of course, they're the middle leaf. Now, on the lower epidermis, frequently on the lower epidermis, you have the mouth of gas exchange in leaves. Because remember, photosynthesis needs to breathe in carbon dioxide and breathe out oxygen. Therefore, you need a mouth with which to exchange these things. The mouth where this exchange is happening is called a stomata, and stomata happens to be the Greek word for, yeah, you guessed it, mouth. Now, in terms of the internal structure of this mesophyll, you'll notice that there are two different seemingly cell layers. One that looks fairly upright, and one that looks like it's got some holes in it. The upright layer we call the palisade mesophyll, or palisade middle leaf, because palisade is another way of saying fence. Yeah, it kind of looks like a fence, right? We're not that clever in science. You will learn over and over again that many of the terms that we use that sound technical, like stomata and epidermis and palisade, are all just different ways of saying a common word in English from another language. So that way we can be more specific. Because if I say fence, you might not associate it with the layer of the mesophyll that performs the highest amount of photosynthesis. Whereas palisade mesophyll, you'll now, hopefully, always think of the fact that you have this upper layer in the middle of the leaf where so much photosynthesis is being performed. Now, because most of, or a lot of the absorption of sunlight is occurring in that palisade mesophyll, the mesophyll layer underneath it needs to be have more gaps in order to get the light that's being absorbed from the top layers. There's not as much light going through. The palisade is casting a little bit of shade on the lower level. And because the lower level has this kind of Swiss cheese appearance, kind of like a sponge, we very cleverly term it the spongy mesophyll. And these palisade and spongy mesophyll layers are all just made of cells. Of course, from our last lecture, you know the basic structure of a plant cell, with its nucleus, its ribosomes, mitochondria. Yes, plants still perform photosynthesis and cellular respiration in the mitochondria, the vacuole, the chloroplast, and all of the other organelles that we went over. Today, we'll take a deeper dive into the chloroplast itself, because it is within the chloroplast that we see photosynthesis happening. So let's take a look at those internal structures in the chloroplast. We, of course, have our double outer membrane, as we discussed a lot last time. And then we have our thylakoids, these individual stacks of membranes, these bags that keep whatever juice is on the inside separate from the very internal structures of each thylakoid. The juice on the outside of the thylakoids is called the stroma. And then the area within the thylakoids is called, very cleverly, an inner membrane space, or the lumen. Now, when thylakoids are arranged in these giant stacks, we call that a granum. Grana for plural, granum for singular. And these kind of operate like solar cells. They stick on top of one another, like this. Light will come in. It'll be absorbed by the first, and then absorbed, 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 absorbed. And each of these are working to convert that light energy into chemical energy in a variety of different steps. Now you'll notice that we are going to continue to keep getting deeper and deeper in this topic with more understanding of what exactly is happening molecularly. So to that end, let's take a look at what are the actual chemical processes that are happening in these thylakoid membranes within the chloroplast. So, within the chloroplast itself, on the thylakoids, you have something called, let's go ahead and write this clearly. you have something called the light reaction. It is the first step of photosynthesis. The purpose of the light reaction is to charge up our batteries. The batteries that fuel the chemical reactions within a cell, particularly these chemical reactions in the plant cell, are ATP and NADPH. I won't ask you to know more about those, other than these are your fully charged batteries. ATP is adenosine triphosphate, triphosphate indicating that it is fully charged because it's got 3 phosphates, and phosphates are currency for chemical energy, just like the dollar is the currency that you use to go ahead and buy lunch, soda, or perhaps the best breakfast in the world, more Raisin Bran Crunch. And the light reaction is going to charge those batteries. Now, in the second step, that's happening in the stroma, the cell juice of these chloroplasts, you have the dark reaction. And the dark reaction is going to use those fully charged batteries to produce sugar. But in producing this higher energy sugar, it's going to deplete those batteries. And therefore ATP will be converted into ADP. Oops. The T standing for 3, the D standing for 2, adenosine diphosphate. So you've lost a phosphate, and therefore you become not charged or less charged. And NADPH is going to convert into NADP+. In this case, the hydrogen and its associated chemical bonds are the source of energy here. So that's the basic overview of what photosynthesis is doing. You have a light reaction that's going to charge batteries. Those batteries are handed off to the dark reaction. The dark reaction is going to use those batteries to make sugar. The light reaction is happening on the thylakoid membranes because it needs a solid surface in order to occur. And the dark reaction is going to happen in the juice, the stroma, because it needs a whole bunch of moving parts. So then you might ask, well, why does the light reaction require those membranes? So of course, we're going to go deeper and illustrate why the light reaction occurs on thylakoids. So let's start by drawing an actual thylakoid. Actually, I'll move this down here so that way we get a little bit more space here. This is the light reaction. So here we have a thylakoid, and on this thylakoid membrane, we have proteins. Many, many hundreds of proteins. Now, a specific type of protein that has color is called a pigment. You might have known it as, well, the things that give color to our shirts or pants or hats. But pigments have this ability to absorb light of specific colors and then reflect light that they don't absorb. So for example, I happen to be wearing a green shirt right now. The pigments in my shirt are absorbing all of the lights that are not green and reflecting back the green light so that way my eyes detect it as green. In the same way, you're going to have pigments in this thylakoid that are arrayed in what kind of looks like a satellite dish. And these individual pigments are going to form these giant clusters together and be absorbing a whole bunch of light energy. You may know those pigments as chlorophyll. Much more on that in a bit. Now, these chlorophyll molecules are found in both of these two different satellite dishes. The two different satellite dishes are very cleverly named P680 and P700 for the wavelength of light that they focus on. And if you happen to know some physics, you might happen to know that there's different energy in light based on the wavelengths of light. But if not, you can go ahead and just keep track of these as P680 and P700, or very confusingly, photosystem 2 and photosystem 1. Now they are named as photosystem 2 and then photosystem 1 because photosystem 1 was discovered first and photosystem 2 was discovered second, but in the actual process, photosystem 2 operates first and then photosystem 1 operates second. You have to have P680 before P700. So remember them numerically based on that rather than their names of 1 and 2. Now, these photosystems made up of a whole bunch of chlorophyll are going to be absorbing light that's coming in. And now, what that light does, let's make that G legible. Kind of, right? That's kind of a legible G, pretend. Now, what that light does is that when it hits that pigment, it starts to heat it up. And the pigment's absorbing that energy in the light. Now, again, if you've taken physics, you'll know that light, or heat rather, is energy of vibration. And so you have kind of a wiggle to each of these pigments. They vibrate, and they vibrate, and they vibrate. And since they're all packed in so tightly in a dish shape, they start transferring the energy of their vibrations to the very center of the dish. You can imagine this as though all of us were packed into a tiny room, and some folks on the edges of the room started to vibrate. The people in the middle would also start to shake a little bit because we're all so closely packed together. The vibration energy transfers. Now, as the vibration energy transfers, you do accumulate more and more energy. And the reason why you want to accumulate that energy is so that way at the very center of this photosystem, in something called the reaction center, you can perform one of the coolest chemical reactions out there. In this reaction center, water is going to be taken in and out of this reaction center. Oops, let's draw this going out. You will release oxygen and those hydrogen ions. So it is at this reaction center that water is being converted into oxygen. So, going back to our equation for photosynthesis, we see that the water is where oxygen comes from. That's really weird, right? Water, the water that a plant drinks up, is going to be converted into the air that you breathe. This is happening at the reaction center in the light reaction. Step 1 of photosynthesis. See how we keep going deeper? Okay, so we've made water. in this first step of the light reaction at the reaction center. But why? Why did we make oxygen? Did we make it for the purpose of making oxygen? Well, as it turns out, it's not for the oxygen that this is occurring. Oxygen just happens to be a byproduct. It's for those hydrogen ions. And ultimately, the electrons that are released. Electrons are a molecular way of transferring energy from one thing to another. So you've essentially ripped off the hydrogens at the reaction center, which I forgot to mention has a very important chemical called manganese. And you'll also hear the reaction center. Let's see. Let's not get ahead of ourselves. Reaction center. Or the oxygen evolving complex, as it is more technically called, the OEC, having manganese, an essential component of being able to rip apart water, because, I mean, think about it. How difficult does it have to be in order to rip apart water into a gas? I'm not talking about heating up water so that way it goes from in my pot on my stove to the air. That's water vapor. I'm literally talking about ripping water apart so that way it becomes oxygen and hydrogen. That's going to take a very specific element, like manganese. Now, the reason why it's being ripped apart, again, is to generate this high-energy electron. You can think of this high-energy electron like a kid in a candy store. We just hopped you up on a whole bunch of sugar, and we're going to utilize that child to generate energy. We're going to do so by placing them on a treadmill. They will hop from enzyme to enzyme. And we'll go over the enzymes in this process next time, but I don't want to go too far too fast the first time around. And there's a whole bunch of enzymes that this electron gets handed off to. You know, this treadmill, then you go on the rower, then you go on the bike. And in the process of doing so, and running on all those machines, that electron is converting ADP into ATP. So we're charging the first of our batteries. After you charge ADP into ATP at photosystem 680 or P680, the electron then gets handed off to P700, which again deals with higher energy light. Therefore, it's going to generate a higher energy battery. That electron gets handed off. We again see light coming in, all the vibrations happening at P700, and then enough of it accumulating so that way that electron again goes sky high, gets handed off to a different set of enzymes, and those enzymes are going to charge our second battery from NADP plus to NADPH. Whoa. That's where it's going? Yeah, that's exactly where it's going. To charge the high energy battery. And yes, NADPH is a higher energy battery than ATP. You can pretend, and it oftentimes is actually illustrated this way, that there is an axis, a graph here, where the higher you go, the more energy you have. So NADPH is a higher energy molecule than ATP, but both of them are going to be necessary in order to run the dark reaction that we're going to go over in a second. Now, additionally, you are going to see that is not just chlorophyll present here in these photosystems, but a couple of other pigments. Things like carotenoids and phycobilins. You may know these types of molecules as antioxidants. Because yeah, plants have a whole bunch of antioxidants, right? You see that on the labels of the food you eat. Blueberries in particular, right? Lots of the food that we eat have large amounts of antioxidants. We'll go over why that's the case, hint, hint, in a bit. But I do want you, I'm layering this like a cake. So you've got the light reaction, where P680 is absorbing light energy coming in, transferring through vibration to the reaction center, or the oxygen evolving complex with manganese, so that way water can be ripped apart, oxygen produced, and an electron shot up to high energy states. That electron then goes on a treadmill, that treadmill being a bunch of different enzymes to charge our first battery ATP, hand it off to P700, where the vibrations occur again because light's coming in. That energy is transferred to the electrons, shooting it sky high. We put that kid on a treadmill again, and enzymes are used to charge up the battery NADPH. Now we know that those two batteries, ATP and NADPH, are going to be handed off to our dark reaction. Or, named after the scientists who discovered it, the Calvin-Benson-Basson cycle. Now, in your textbook and in other resources, you'll oftentimes see it called the Calvin cycle, sometimes for shorter. It was originally called the Calvin cycle, but that did not give fair credit to the individuals who truly discovered it. Benson and Basson were instrumental in this work, and Calvin, as their boss, was happy to take a lot of credit for them. I do incorporate this story in any class I teach because I happen to be taught by one of the students of Benson and Basson. They went on to have scientific careers as well. And while Calvin originally got the Nobel Prize, it was later shared with Benson and Basson because their work was instrumental. In fact, Calvin originally threatened to fire them if they performed the work because he thought it was so crazy. They did so behind his back and therefore discovered the basic premises of this process. When he saw that it worked, well, Calvin was ready to take some credit. Maybe not the best legacy to leave behind. You have jerks in all levels of society. Or at least that is the story that I've heard. Now, in the Calvin-Benson-Basson cycle, you are going to have, just like in the light reaction, you are going to have enzymes that are driving this process. But I will introduce the one enzyme that I expect y'all to know for the dark reaction here. That enzyme is called rubisco. Rubisco is incredibly important, and we'll spend time today talking about it and next time talking about it, because it is an imperfect molecule. But rubisco is the enzyme that's responsible for grabbing carbon dioxide out of the air and bringing it into the Calvin cycle. And it's called a cycle because it's able to repeat. You're able to keep running this cycle over and over and over again in order to continually make sugar. These plants, of course, want to continually make sugar. Rubisco is named Rubisco not because it rhymes with Nabisco, and Nabisco makes cookies, and cookies are made of sugar, and Nabisco cookies are great, but because of the things that it works on. Rubisco works on a molecule called RUBP, or ribulose 1,5-bisphosphate. I don't expect y'all to know that, but I'll write it out here. Ribulose 1,5-bisphosphate. So you see the RUBP. This happens to be a 5-carbon sugar molecule, as anything with ose in biology is going to be a sugar molecule. Think cellulose, glucose, ribulose. All of those are sugars. Now, because Rubisco works on ribulose 1,5-bisphosphate and has both a carboxylase and an oxygenase activity, these are two different enzymatic properties. We call it Rubisco. So it's going to be working on this molecule and having these properties, and therefore we name it after that. This is the only molecule that I'll expect, I'll even introduce this level of detail for. But I just want to give you an idea that just like with the palisade mesophyll and the spongy mesophyll, scientists really do try to name things after what they do. But sometimes those functions are incredibly detailed. You won't need to know what carboxylation or oxygenation is up until perhaps your 4th year of undergrad in a bachelor's degree in biochemistry. So we'll just leave them as names here as to exactly what's happening. But to give you an idea that these things should, in theory, make sense. They are not arbitrary. So carbon dioxide is being grabbed in through Rubisco into the Calvin cycle. And now going back to our equation for photosynthesis, we know that carbon dioxide is an input, and we've already used up the oxygen as an output, so therefore we know that carbon dioxide is going to be produced into a sugar molecule, a glucose molecule. That's right. The carbon dioxide that a plant breathes in is going to be converted into the sugar that you and I eat. That's pretty nutty. If that isn't magic, I'm not certain what is. Air goes in, a sugar molecule comes out. But this molecule is not yet a glucose. It is instead a 3-carbon sugar. Glucose is a 6-carbon sugar. Therefore, this Calvin cycle is going to need to cycle twice in order to produce 2 3-carbon sugars that then join together to produce a 6-carbon sugar. And glucose is one of the major currencies that sugar is trafficked in. Just like you have dollars moved around in single dollar bills or $5 bills or $10 bills, our chemical energy is moved around in glucose. So glucose is the equivalent of a single dollar bill. But you've also got a hundo, a $100 bill. When you have a lot of glucose molecules, you convert them into a starch molecule, chemically known as amylose. So we've discussed the outputs of the dark reaction. Carbon dioxide is coming in, a 3-carbon sugar is coming out. You perform the cycle twice for a 3-carbon sugar to join with another 3-carbon sugar to make a 6-carbon sugar. And then when you've got a lot of 6-carbon sugars sitting around, you go ahead and convert those 6-carbon sugars into these giant chains of amylose or starch, like you have in starchy potatoes. Love French fries, right? You will find I make as many connections to food as possible because I love food. Now, we also know that part of this process of the dark reaction is going to be to deplete the batteries. ATP and NADPH are going to come in, and they're going to get used up. And out will come ADP and NADP+. So that way, those products can go back up to the light reaction, and we'll do it all over again. This process is happening over and over again. Now, this depletion of energy from ATP and NADPH is a step called reduction. It's the second step of the dark reaction, or the CBB cycle. The first step was carbon fixation, done by Rubisco. Just forgot to mention it. So step 1, Rubisco grabs CO2, puts it on ribulose 1,5-bisphosphate, or the backbone of this cycle. In the process of fixation, energy is added to that carbon dioxide from ATP and NADPH, and a sugar molecule is released. Now, this whole process of adding energy, zapping the backbone, and releasing a sugar molecule damages the structure of this backbone. You need to then go through your third step, called regeneration, in order to run it back. Because remember, you need to perform this cycle 2 times in order to produce a single glucose molecule. And glucose is that $1 bill of chemical energy storage, of sugar energy storage. So in the dark reaction, we have carbon dioxide coming in, being grabbed by Rubisco in the process of fixation. ATP and NADPH are discharged of their energy in the process of reduction in order to produce a 3-carbon sugar. And then the mangled byproducts of that are regenerated in step 3, regeneration, in order to be able to run the cycle back again. And in this way, you have the 2 steps. The light reaction happening on our thylakoid membranes, where electrons are being used to charge batteries. Those batteries then go to the stroma, where things are floating around and bumping into each other. Rubisco is bumping into carbon dioxide. ATP and NADPH are bumping into the backbone. Sugars being released, and molecules are being regenerated. And in this way, you're able to cycle through this and continually make sugar in this photosynthesis reaction. Kind of cool, right? Now, this is kind of the backbone of what we'll be discussing. We'll end today's lecture by diving a little bit deeper as to what is going on with those different forms of chlorophyll and the light that's hitting our thylakoid membranes at the photosystems P680 and P700. So, light itself is made up of all different kinds of colors, and even things that to us don't look like colors. You have radio waves that you listen to radio through. You have infrared light. You have your visible spectrum of light that has all the colors that we detect. Then you have UV light, or ultraviolet light, X-rays, gamma rays, that are all different photons that are coming from the sun. And it's weird to think that these pigments, that we only see a small portion of the entire spectrum of light out there. Thank goodness we're not seeing radio waves. Otherwise, you'd literally see the colors of the radio. Interesting, right? Now, these colors matter because they explain why plants are green. And that's what we're ending today's lecture with, an explanation of why plants are green. We have in plants several different chlorophyll pigments. We have chlorophyll A, chlorophyll B, and these are found in all plants, but also in algae, with chlorophyll A being found also in cyanobacteria, which you might know better as blue-green algae. They're not eukaryotic, they are prokaryotic, but they are also able to perform photosynthesis. Therefore, we think chlorophyll A happens to be the original version of this. Chlorophyll B is an innovation. And what you'll see based on this graph is that chlorophyll A specializes on absorbing light that's red and light that's purple-blue. So we see the absorption peaks of chlorophyll A at the red and the purple-blue side of things. Chlorophyll B absorbs preferentially the blue/teal and a little bit of the orange. And so now, if these two pigments are absorbing the purple, blue, orange, and red colors, what's left over? What colors are not being absorbed? Well, green. And it's because these pigments are absorbing all the colors except for green in order to perform that vibration energy transfer that you see plants as green. There are other chlorophyll pigments that do absorb that green light, like chlorophyll C. Chlorophyll C absorbs green light, and therefore the things that have chlorophyll C are not green. They are brown algae, like kelp. And so if you have no more of this green spectrum, what do you have left? The yellow-brown spectrum. So yeah, that's why brown algae are brown, because they have something that's utilizing that green wavelength of light. Kind of cool, right? We also have these accessory pigments like carotenoids and phycobilins that absorb, sometimes this more orangey color of things. And obviously are named in part because of the different spectrum that they reflect. Carotenoids are absorbing the greenish and bluish side of things, and therefore are reflecting your reddish-orange, like orange carrots. But their real purpose inside of the P680 and P700 photosystems is to be antioxidants. Now, oxygen, when breathed in your lungs, can be used to make energy through the process of cellular respiration. But that is in its O2 form. Oxygen that's made here at P680 is in its O1 form. And when it's in its O1 form, it has a -2 charge. That means it's looking to rip other molecules apart in order to balance itself. When it finds that second oxygen, it's able to be more stable, but by itself, it is fairly unstable. And because it's unstable and ripping everything apart, potentially causing damage, you need something in those photosystems to take the hit of that damage. That's where our antioxidants like carotenoids and phycobilins step in. Their whole purpose is to take the hit for that damage of those free oxygens. They take the hit so that way your DNA doesn't have to. Kind of cool, right? And so that's where we'll end our lecture today. Having gone over the basics of photosynthesis, the light reaction, the dark reaction, all of the steps necessary, where in the leaf this is occurring, and even some of those accessory pigments like carotenoids and phycobilins. And next time, we'll take an even deeper look at some of the enzymes involved in these processes, particularly the light reaction, talk about what happens when photosynthesis goes wrong, because it does, and talk about some of the evolutionary innovations on photosynthesis, because those exist too. See y'all next time.